Hi, everyone. My name's Meharvan Singh, and most of you know me as Sunny, and I'm very pleased to be part of the 5th International Symposium on Sigma-2 receptors. And I want to first and foremost thank the organizers, including Cognition Therapeutics, for bringing us back together. And today I'd like to share with you some work that we've been doing related to PGRMC1, a presumptive important component of the sigma-2 receptor, and its role in progesterone's protective effects, particularly focused today in discussing how progesterone through PGRMC1 mediates the release of a neurotrophin called brain-derived neurotrophic factor, or BDNF. Just a couple of quick disclosures to share before I get started into the science. I wanted to start today with this slide as it illustrates how we kind of got started in this work. Many years ago, we asked some questions trying to understand why women had a higher prevalence of certain neurodegenerative diseases like Alzheimer's disease relative to men. So the idea that emerged in our thinking and that of others was that perhaps the very significant and precipitous decline in circulating gonadal hormones, which were emerging as having important effects on the brain outside of just reproductive function, maybe that loss of those important hormones rendered the brain vulnerable to certain neurodegenerative diseases. And so we asked the question in an animal model whether ovariectomy of these animals would result in changes in their cognitive function, their neurochemistry, and we looked at the cholinergic function in particular, and also neurotrophin expression. And consistent with the theme of this presentation, we focused on brain-derived neurotrophic factor. And what you're looking at here is in situ hybridization for BDNF, brain-derived neurotrophic factor, and the noting that the expression of BDNF was profoundly reduced in ovariectomized animals. These are animals that have been ovariectomized for about 7 months. Importantly, we noted that indeed estrogen could at least partially restore the expression of BDNF, noting here the reemergence of signal in the hippocampus. So that paired with this understanding of the biology of the menopause, where the average age of the menopause in the United States at least is 51, and the mean lifespan for women is about 81, the question we asked was, what does it mean to spend about a third of one's life in a hormone-deprived state? And as such, given what we had started identifying, consequences of ovariectomy on cholinergic function or neurotrophin expression, we started to ask the provocative question of whether or not this reduction in ovarian steroid production could render the brain at risk for certain age-associated diseases, to include stroke and Alzheimer's disease. And whereas we and many other laboratories showed lots of important effects of estrogen on the brain outside of the typical confines of reproductive biology, we started to also recognize that when you ovariectomize an animal, or when a woman goes through the menopause, from an endocrine perspective, that represents a decline or loss of not only circulating estrogen levels, but also of circulating progesterone levels. So is it possible that this rather significant reduction in BDNF expression could have been attributed also to the loss of progesterone? And so our lab and many others embarked on asking questions about whether or not progesterone in and of itself could be protective. And our lab showed quite a number of years ago that progesterone can in fact elicit the activation of the ERK-MAP kinase pathway, as well as the PI3 kinase AKT pathway. Indeed, progesterone could increase the expression of BDNF. And importantly, to kind of connect the dots, that these signaling pathways were in fact required for progesterone to protect cortical neurons from such insults that simulate age-associated diseases like oxidative stress. While we did find that progesterone could increase BDNF expression, we wanted to understand what was the cellular molecular mechanisms underlying that. So we started looking at what receptors are involved. And what we found is that as a first pass, that the classical progesterone receptor does seem to be critically important for progesterone-induced BDNF expression. And this is an experiment done in cortical neurons where progesterone increased BDNF expression, but the pharmacological antagonist, RU486, blocked that quite nicely. We recognize that RU486 isn't the cleanest of pharmacological antagonists to the progesterone receptor, so we did follow up with using progesterone receptor knockout animals. And here what you can see is each bar represents the effect of progesterone. So in wild type animals, progesterone increased BDNF relative to the control set at 100%. But progesterone receptor knockout animals, progesterone had no effect on the expression of BDNF. And so we were confident that the classical progesterone receptor was really quite important. So that resulted in the emergence of our working model that said that progesterone acting through intracellular progesterone receptors regulated the expression of BDNF. But we started to appreciate that just regulation of BDNF expression and thus increasing the intracellular buildup or pool of BDNF was really not going to be sufficient to promote protection of the system, i.e. the brain. And so we started to turn our attention to what governs BDNF release. Does progesterone have a role to play, or is that some completely independent mechanism? And so our attention turned to PGRMC1, which later we came to learn has an intimate relationship with the identity of the sigma-2 receptor. And what I hope to convince you through the next couple of slides is that progesterone interacting with PGRMC1 elicits the activation of a member of the MAP kinase pathway, ERK5 signaling, that then is key to the promotion of BDNF release. And the inference is once you get BDNF release, then it can act through its cognate receptor and it can promote such brain promoting effects that include synaptogenesis and indeed cytoprotection or neuroprotection. So one slide to introduce this relationship between progesterone and BDNF release is here, where I show that progesterone not only resulted in an increase in BDNF release, but that when we knocked down PGRMC1 using siRNA, that effect of progesterone was lost. So here again is the induction of BDNF release in response to progesterone, and then in the presence of the PGRMC1 siRNA, progesterone receptor was unable to elicit BDNF release. Well, what we wanted to kind of learn was, is progesterone-induced BDNF release mediated by just PGRMC1, or is the classical progesterone receptor also involved? Well, as one approach, we again used RU486, the pharmacological inhibitor of progesterone receptors, to test that question. And what we found was RU486 was incapable of blocking the effect of progesterone on BDNF release. But what we were kind of intrigued with is the fact that U0126, which is an ERK, classically referred to as an ERK1/2 inhibitor, completely abolished the effects of progesterone on BDNF release. So we started to explore this a little bit more, and we appreciated the fact that U0126, if I can come back to that slide real quick, not only inhibits the ERK1/2 pathway, but also inhibits ERK5. So we asked the question, well, which of those two signaling pathways is more important as a mediator for progesterone-induced BDNF release? And what we found was only ERK5 phosphorylation was reduced in terms of progesterone's ability to elicit ERK5 phosphorylation when you knock down PGRMC1. So here what you have is progesterone alone increasing the phosphorylation of ERK5, and in the presence of the siRNA, so when you knock down PGRMC1, progesterone was no longer able to elicit ERK5 phosphorylation. In sharp contrast, progesterone here elicits ERK1/2 phosphorylation that had been reported by our lab and many others. But when you knock down PGRMC1, progesterone still continued to elicit the activation of ERK1 and 2. So this suggested to us that ERK5 was really the key MAP kinase pathway that was involved in eliciting BDNF release. Well, to test this further, we wanted to see whether or not knocking down ERK5 specifically would abolish the effects of progesterone on BDNF release. And the short answer to that is yes. So here again, relative to control, we have progesterone eliciting BDNF release. And then in the presence of the ERK5 knockdown, or the siRNA, progesterone was incapable of eliciting BDNF release, that is. So does that have anything to do with the promotion of cell viability? That is to say, progesterone working through PGRMC1, eliciting ERK5, releasing BDNF. Does that have any implications to that which arguably counts, and that is the promotion of cell viability? So one experiment that we did was to determine in progesterone receptor negative but PGRMC1 positive cells what the effect of progesterone was in protecting against a metabolic and oxidative insult called IAA, or iodoacetic acid. So here you can see iodoacetic acid reducing the viability of the cells, and progesterone at concentrations that are parallel to what we would interpret as physiological really had no effect. In contrast, when you had both PGRMC1 and the PR present in the cells, so these are PR transfected cells into those PGRMC1 positive cells, now progesterone at those same concentrations promoted protection, suggesting again that the 1-2 punch is important. You need a mechanism that not only governs the expression of BDNF, but you need a mechanism that promotes its release. And so the emergent working model then was that progesterone working through the classical receptor elicits BDNF expression, and then through PGRMC1 and an ERK5 signaling pathway, BDNF can then be released, and after that act on its cognate TRAC-B receptors to elicit those features, characteristics that would promote brain health, function, and viability. So we wanted to see, though, what regulates PGRMC1. Now that we know that PGRMC1 is so important in regulating BDNF release, at least in response to progesterone. So we turned our attention to some microRNA, and to kind of cut to the chase, in the interest of time, I'll share with you that we focused on the LET7 family. And what we found was that progesterone's effect on BDNF release was completely abolished when we increased the expression of LET7I. So LET7I appears to be a negative regulator of progesterone-induced BDNF release. What we found was that increasing LET7I reduced PGRMC1 expression. Given that we placed an importance on PGRMC1 as a key mediator of progesterone-induced BDNF release, and therefore progesterone-induced protection, we viewed that LET7I, increases in LET7I, would dampen any protective effects of progesterone. So conversely, we asked the question, if we inhibited LET7I, could we make progesterone's protective effects better? And so we chose to explore this in an animal model of stroke, using the middle cerebral artery occlusion model. And what you see here are the 4 representative groups that we looked at. the control, the non-stroked animals. And what you're seeing here is TTC stained coronal sections of the mouse brain. And where you see the red, that's where you have metabolically active tissue. And where you see white, that's where you have ischemic tissue. So following the stroke with no treatment, we get a nice reproducible stroke. When we just applied progesterone, we didn't have much of a protection at all, as you can see. But when we co-applied progesterone and this LET7I inhibitor, we got a nearly complete protection. This was really underscored by this video that I'm going to show you, where here on the left, these are animals recovering from the stroke surgery 1 day after treatment with progesterone alone. And focus your attention on this middle animal, and you'll see this very sort of stereotypic behavior of circling in one direction, because the animals are weak on one side. They have hemiparesis, and so they have dominant muscles on one side, resulting in unidirectional circling. But in animals where we treated them with the LET7 inhibitor, we saw a rather remarkable recovery in a very short amount of time. And in fact, behaviorally, we couldn't distinguish these animals from the wild type animals, non-stroked animals. Extending this analysis a little bit further, what we did was we looked at the peri-infarct area in humans that would be equivalent to the penumbra, and looked at synaptophysin expression. And what we found is that, so here again, we have, as a function of time post-treatment and surgery, that synaptophysin in red is nicely expressed in the non-stroked animals, in that region of the brain that would be equivalent to where we see the peri-infarct in a stroke. In the stroked animals, we see a pretty significant reduction in the expression of synaptophysin. That seems to be at least partially recovered over time with progesterone alone. But remarkably, at a fairly rapid rate, the co-application of the LET7I inhibitor resulted in a much faster recovery of synaptophysin expression in these animals. We extended this analysis to in vitro to more tease out the mechanism and the role of LET7I. And here what we see is progesterone increasing synaptophysin expression. And when we applied LET7I, recall that LET7I is that dampening influence on progesterone's protective program by reducing PGRMC1. Progesterone's influence on synaptophysin expression was lost. I should point out that in just stroke alone, when you just induced the stroke in the animals, LET7I actually goes up, which means more to the potential for dampening that LET7I, so PGRMC1 expression can be restored, and so progesterone can elicit its protective program. Well, finally, I wanted to share with you the downstream consequence of releasing BDNF. And the experiment that we did was to determine whether or not conditioned media from progesterone-treated cultures would in fact elicit its protective effects through TRAC signaling. The TRAC-B is the neurotrophin BDNF neurotrophin receptor. And so supporting the transsynaptic protective program that we're really talking about here. What you're seeing here are cultures treated with hydrogen peroxide. Here is in the control group, where you have a certain number of apoptotic nuclei that emerge in this field of view. The blue is the Hoechst stain for apoptotic nuclei. And then in response to conditioned media from progesterone, you get a very nice protection. In the control groups, we see a little bit of protection from this conditioned media because there is some constitutive release of growth factors and neurotrophins. But what I want to point your attention to is that when we applied the inhibitor for neurotrophin receptors to that environment where we have progesterone induced conditioned media, So you have one group of cells, you treat them with progesterone, you get the conditioned media, you apply it to neurons, and you look to see whether that protects. That's this group, and it did. But now, if you block the neurotrophin receptor, would that conditioned media still protect? And the answer is that it didn't, supporting the idea that indeed this progesterone-induced BDNF release is being mediated through the neurotrophin receptor. So to wrap up, what this body of work has yielded to us is that you need both the classical PR and PGRMC1 to enable optimal progesterone-induced protection. And it also speaks to the critical importance of BDNF release as an important mediator. And finally, that LET7I appears to be an important regulator of PGRMC1 that in turn modulates progesterone's effects. So I want to acknowledge the lots of collaborators, lots of smart people that I have the good benefit of working with, our funding agencies, and of course the people who've done the work. I have transitioned to Loyola University Chicago in the last year or so, and I want to acknowledge the new lab members who are continuing the pursuit of these questions. I thank you for your attention, and we'll look forward to taking questions.